Okay. Good afternoon, everybody. Okay, I'm sure you enjoyed your lunch. And for any seminar, any event, the most challenging part of the seminar is after the lunch. Okay? Because everyone is feeling sleepy, tired, waiting for coffee break. So to avoid that, we have something more interactive. I am sure that you will enjoy. Okay? So what we are going to do now is, as you can see the instruments around you, and some of you are already visited, but it may not be enough time for you to visit to each and every instrument. So what we have today for you is the experience workflow session. May I have the slides, please? Okay, I think they are still in lunch time. Okay, back. So, in experience workflows, what we are going to see is how the pharma workflows today and how they may be looking after a few years in our workflow zones. Okay. So, in any pharma company, when you have routine workflows, okay, starting with chromatography and spectroscopy, also the balance and chemistry. You also have advanced workflows with MS now, wherein the regulations for impurities are increasing. These advanced workflows are getting more common now. Okay? So we will have some advanced workflows, and the most important compliance part with our informatics solution, which combines all these instruments together. Not only that, but we also have some futuristic workflows, like some new instruments and the synergy. As you know, Shimadzu is the only company having medical and analytical devices, and we can hence provide more healthcare solutions. Okay? So these are the futuristic workflows. We have all these workflows around the corner here, but you don't have to do anything. You can just be where you are, and I will walk you through all the workflows, and we will explain what we have today. Okay? And afterwards, you can have some time to go to the booth and ask any questions that you have. Okay? So I'm sure this will keep you awake and agile. Okay, so let's walk to the first experience workflow, which is the routine workflows. Okay, and to explain this workflow, I have with me David Chan. You have just seen him receiving the award. He is a product manager and also pharma market in charge. Okay, so David, what do you have today?
Thank you, Sandy. Thank you, Sandy, again. Ladies and gentlemen, my name is David, and in fact, today I feel very honored to be able to stand in front of so many pharma delegates here to present to you what solutions we have specifically for routine workflow. Now, a blockbuster medicine will not just appear overnight without a efficient, effective, successful R&D drug discovery process. As what Sandy has also mentioned, a drug discovery process is a long process. It takes at least 14 years to get a blockbuster drug out. So this is a long and tedious process, and how Shimadzu, we can help you in making this tedious process a smooth one. So before lunch, we have already witnessed and experienced the new benchmark of the award-winning new Nexera series, the first AI-enabled and IoT-driven smart analytical instrument. Essentially, this instrument is powered by analytical intelligence. The whole concept is that the instrument want to deliver is to free the chemists away from a lot of repetitive work so that they can spend more time doing higher level work such as designing molecule, data analysis. So as a whole, the new Nexera series is the best in class UHPLC, is the best in class application system, and it's the best in class front-end LCMS MS. Now, purification is also one of a very important topic of the whole research and development process. So the new Nexera series can also be configured together with our latest liquid handler, LH40, and it can be easily configured as a standard preparative system, as an advanced mass-directed preparative system. You can also even upgrade the system to have a higher level UFPLC, or in full we call it ultra-fast preparative and purification liquid chromatograph. I was always joking with Sandy is that I wish to bring the UFPLC back home because what? As an organic chemist, I was an organic chemist before. When I did preparative work, I need to spend a lot of time removing aqueous solution. And UFPLC essentially can remove all this trouble that I have in shortening the whole purification process. Quality control, probably most of you sitting in this ballroom must be thinking this is a very heavy topic and is most of the biggest concern for all of you. So we are here to remove all the concerns because we have our LC and GC, the proven industry standard LC and GC, so that for quality control. Both instruments are innovatively designed and engineered with full automation process so that you can fully enhance your laboratory efficiency. Spectroscopy. Shimadzu spectroscopy product, I don't need to further elaborate. It has been so popular. But one key point that probably we always overlook is the data integrity that produced by spectroscopy product. So we can even safeguard you by providing our very unique spectral network solution so that all the data produced by Shimadzu spectroscopy product, you can be configured and integrated in a compliant network solution. We have excellent instrumentation. How about the core of chemistry? Our very own core focus, the smart choice in consumables, provide you a wide range of consumable products, right from a separation column to a MS certified valves to a high performance UV cuvettes. So ladies and gentlemen, this was just a very brief introduction of what we have. Essentially, we have more than what we've shown here. So I will encourage you, if you want to know more, later on we can have more interactive session. Now I would like to pass the mic to my colleague Jackie to share with you more of what he has for the advanced workflow. Jackie, please.
Thank you, David. Hi, good afternoon. My name is Jackie. As the pharmaceutical industry is transforming, the regulation is getting more and more challenging by the day. As such, advanced instrumentation like the MS is gaining more and more popularity because it is required for the trace analysis of various impurities. So, let me see where is the next slide. Okay, it's working. So, when it comes to potential genotoxic impurities, GC-MS-MS is a very powerful instrument for highly sensitive analysis of compounds like the NDMA and NDEA, nitrosamines in certain drugs, one of the hottest topics in the pharma industry right now. We can use the headspace trap for analysis of these compounds with minimal sample preparation. And in fact, we can also use an auto-liquid sampler to have wide-range analysis of various impurities that can be extracted from the API or from the drugs. In fact, we can combine them, as you would see on the slide there, that you can actually have both the auto-sampler for liquid and the headspace in a single instrument. So you can use it as much as you require. The LC-MS-MS is another instrument that can be used for this analysis for its short analysis time, and on top of that, it is capable of analyzing some compounds which are less compatible with the GC. For the extractable and leachables in packaging material, GCMS proves to be a very useful solution because it is able to identify compounds by matching it against a library. We can use the headspace trap or thermal desorption, these accessories which can be attached to the instrument, so that we can directly put the packaging material into the instrument for analysis. So essentially, no sample preparation is required. And alternatively, we can use the QTOF for untargeted identification of ENL compounds because it is having a very supreme mass accuracy. When it comes to clinical trials, LC-MS proves to be a really useful tool. From the front end that has been described by David, powered by analytical intelligence, to the optimization by using the Connect software and data processing by using the Insight software, the entire method development process in clinical trials is automated, fast, and fully compliant. And if you would like to increase the throughput, you can also have the multiplexing system, in which the dual stream technology allows us to have two analytical lines in the same instrument. So when the first analytical line is ongoing on the run, the other line will start preparing for the sample. And once the first run is finished, you can switch immediately to the second run, thus doubling the throughput. Biopharmaceutical is becoming the next pharma. Shimadzu offers complete solution ranging from the tabletop MALDI, which is perfect for rapid and high throughput analysis of intact protein and glycopeptides. And the LC-MS is a really powerful solution for clinical research and process development with Shimadzu's unique solutions like the N-small and cell culture profiling. And the QTOF is perfect when it comes to complete characterization of biosimilars. And lastly, not forgetting the elemental impurities analysis, which the allowable limits are specified in the ICH Q3D. Shimadzu's ICP-MS can measure these elements with high sensitivity, and at the same time, as mentioned by David about the spectroscopy network, the analysis is fully compliant with the FDA 21 CFR Part 11. Now, having heard so much about the lab network, let us experience the smart lab. Satish, the mic is yours.
Thanks, Jackie. Good afternoon, ladies and gentlemen. Myself, Satish here, and yeah, I'm here. The smart guy near Smart Lab. Welcome to the Smart Lab, which we want to walk through smart workflows. Why are we here? We had seen video in the morning. The purpose of Pharma Summit is all about network, share, and discuss. And we have seen routine and advanced workflows. There are different types of instruments here. And also we have many other varieties of instruments. But the important factor is networking, which I would like to do, which I'm doing it. So in pharmaceutical company, we had a good session in the morning about data science, which is very important and very interesting. And there are keywords like data integrity, automation, and we also talk about analytics, and as well the APQR. And all these data science technologies or requirements which can be addressed by the Smart Lab. So that is what we want to walk through here today with all these display of different types of instruments. We have seen different types of systems, like chromatography data systems that connects with HPLC and GC systems, which is essential. But today's data integrity is not just HPLC and GC. It's all about all the instruments, all the data that is produced by every instrument in the laboratory. So that's where Shimadzu developed Lab Solution CS, which connects not only chromatography instruments, but also spectro and as well MS, and integrates all other laboratory equipments, analytical equipments for a data integrity requirement. And we don't call our Lab Solution CS as a CDS. We call our Lab Solution CS as a analytical data system, because we connect, we control all analytical instruments. The transformation of pharma is all about automation. we're all looking forward to have automation, right? And when we talk about automation, we refer to Pharma 4.0, we can refer to Industry 4.0 and other terminologies. But when we are talking about pharma, the key part of automation is laboratory automation, which integrates different type of systems, different type of instruments, be it as a multi-network, be it as a multi-instrument, or be it as a standalone systems, whatever it is. The automation of all these systems to a single platform is what we are doing today. And if you notice here, we have a beautiful Lab Solutions CS Network ADS, which is integrated with LIMS for complete QC workflows to address all your departments like microbiology, your QC, typical QC, and all other departments. And we have quality management system that regulates your QA workflows. And training management system and BRMS will walk through management and manufacturing requirements. The DMS is the first step towards digitalization. And the entire combination of these smart lab products will lead you to a automated laboratory or laboratory automation. Morning we had about paperless lab is dream or reality. But the final conclusion is paperless lab is essential today. And it is essential, but it is available here. The paperless lab can be achieved with the smart lab, which is a complete paperless laboratory solution with a single platform, a single vendor, and integrating all your processes. And of course, we cannot forget about the mobility, which everyone is carrying smartphones. So we do have smart devices where we can control, we can operate from iPad, all your software, your data, and you have complete hold of your laboratory wherever you are traveling, wherever you are in the world. So that's where the smart lab is scalable from a small system to the large system, is easy manageable by integrating different platforms, and is completely automated, regulated, and it gives you a complete total compliance. So I wish you will visit and have a complete walkthrough of our smart workflows, and let's hear about the future workflows, and I pass the mic to Mr. Daryl.
Hey, good afternoon to you, and thank you, Satish. My name is Daryl. I come from the future. The future is here, and the future is now. In Shimadzu, we are taking you to the future with our futuristic workflow. And first, let's look at this. The little box here, if you can't see, it's here. The little box here is like me, just little box here, is the world's first digital wave ion trap. It's very compact in size. It just occupies the footprint of an A3 size paper. For those at the back who can't see, I'll just lift it up for you to see. So just like the man on the slide, you can just literally lift it up with just one hand. And compared to our floor standing and the bench top Maudi, we have shrunk the size of our Maudi from nearly 2 meters to just 32 cm. And even with this compact size, it's highly sensitive. It can perform MSN. And it has a powerful data processing tool, especially useful for omics research. For example, targeting the compounds like proteins, oligonucleotides, lipidomics, or carbohydrate samples. So next-gen technologies, as you can see here, we have the next-gen GC and the chip LC. They are our future innovations, but I'm showing you here today because all of you are our prestigious guests. The next-gen GC. The next-gen GC uses the plate column, as you can see on the slide. So you will no longer see the 30-meter or the 60-meter GC columns hanging in your GC oven. So at one time, you can put in 2 plate columns, and it can do very accurate hard cutting experiments, meaning you can accurately analyze just the pigs you want to analyze. And of course, AI enabled. It can do self-diagnosis and also automatically check for the gas leak. So for the chip LC, as the name suggests, the separation is on the chip column. So where is the chip column? The chip column is here. I'm taking out the chip column. So this is the chip column. The separation is done on the chip column, and the size of the chip column is customizable for fast separation. So it'll be very useful for rapid screening. So the installation of the column is very, very simple. You just need to slot it in any direction, and it's done. So chip LC. So in healthcare, Shimadzu is the only company that can offer futuristic and synergistic workflow to translate the analytical and medical technologies to advanced healthcare. As you can see on the slide, the diagram shows how the analytical instruments and the medical devices synergize to provide a total and complete solution to the entire clinical workflow, from early disease marker detection to the treatment and to the post-treatment prognosis and monitoring. So for instance, Sonia Vision, the medical imaging device, as well as the LCMS and the DPIMS can be used to detect disease biomarker. For the treatment, we have the Trinius to visualize the disease blood vessels, as well as the light vision in guiding the surgery. For the analytical device, analytical instruments, we have the IM scope, and you can also see the IM scope on the slide. It's a combination of the optical microscope with the mass spec that can be used to localize the distribution of the drugs in the diseased tissue or the affected tissues. So for the post-treatment prognosis and monitoring, therapeutic drug monitoring is very important, and we have the CLAM here, also on the CLAM on the slide. CLAM stands for Clinical Laboratory Automated Sample Preparation Module, and is unique from Shimadzu only, that can be seamlessly connected to Shimadzu LC-MS. It's a fully automated sample preparation module to prep for the clinical samples, for example, your plasma, serum, whole blood, or even urine samples, and it's fully automated. So the purpose and the ultimate goal of this synergism is to optimize the treatment, to improve the clinical outcome, and to better healthcare. So this is our futuristic workflow. Once again, together with Shimadzu, we can create a better future for all. I'll pass the mic to Sandy, the present. Okay. Thank you.
Welcome back to the present. Okay, so I'm sure you must have enjoyed this walk away with all the workflows. And since we have very creative people here, our David has prepared one very short video to wrap up whatever we have spoken for last 20 minutes. Okay, so let's have a quick look at the video. Thank you very much. And okay, so now you have about 20 minutes that you can go around and look at your booth that you want to experience. And you can come back after 20 minutes, okay, so that you can have some walking around. If you are bored, you can check your emails as well, but I'm sure all of you will definitely visit some of the workflow zone, okay? So see you after 20 minutes. Thank you very much. Ladies and gentlemen, thank you very much for your very active participation. And I saw that although everywhere the crowd was there, but the maximum highlight was the futuristic, okay? So all are more interested in future. And that is where, again, we have our synergy. As you have seen in the video, we are the only company who are thinking about the healthcare solution, not only pharma, but going one step ahead. And synergy will be very, very important in our coming products and coming technologies. Okay? So you all deserve a clap for this active participation. And as well as you also deserve a gift, right? So I think most of you have received your gift. If not, it is on your table, okay, in the black bag. Okay, and you will be interested to see, I saw somebody opening as well. So it's a very unique gift hamper for you. May I have the slide to show what we have inside this box? Okay, so we have some products in this bag, such as some chocolates, a toothbrush and toothpaste, tiger balm, essential oil, most importantly, tiger beer. Okay? So it's a very interesting gift. What is unique about this gift is basically all these products in the box are manufactured in Singapore and tested using Shimadzu instruments. A big round of applause. And yes, now the slide comes. In fact, the manufacturer of the tiger balm is here today with us from the Haupar company. May I know where you are? Thank you very much for your support, for sponsorship for this gift. Thank you, sir. Okay. So I'm sure you are enjoying this and you are very tempted to open the box, especially the tiger beer. But please don't enjoy now. We have more interest. Oh, we also have a halal version, sorry. Those who are halal, don't worry. You have a coconut water, which is also tested using Shimadzu LC, okay? And that is where you have a pamphlet inside which explains which instruments were used for testing these items, okay? So we have now another session which we are starting now, but before that, request from some of the audience that taking pictures is interrupting to see the slides. In fact, we are going to send you all the presentations and photographs in a DVD after the summit. So it will be better if you can just focus on and enjoy the talks, and you will get the presentation later, okay? So with that, we are now starting our last session, which is Technology of Therapeutics. And to moderate this session, I would request Dr. Scott from Shimadzu, USA, all the way to moderate this session. Scott.
Thank you, Sandy. So good afternoon, everybody. As Sandy mentioned, my name is Scott Cousdale, and I'm here from Shimadzu Scientific Instruments in Columbia, Maryland, USA, which means I took 3 flights to get here. So this next session is the most exciting session because it is truly global. If you look at the agenda, there are 6 countries represented, and we're actually going to mix things up a little bit here. We're going to start with China, and from China, we have Dr. Seihua Liu, also known as Chloe, and she is the Senior Vice President of Biothera Solutions. So please, let's welcome Chloe.
Thank you, everyone, and thanks the organizer of the Shimadzu and organizing this wonderful meeting. I'm so honored to be here. Well, we were just experiencing a wonderful session and for the next generation and the future workflows and coming from the Shimadzu breaking technologies. So from this morning's technology of the data science, the technology of the tools, and right now we're talking about the technology of the therapeutics. So what analytics technology can do for the therapeutics. So because all my years, all my experience is working on the biologics. So I was so happy to get this title, the presentation for the biopharma. Is that the next generation, next pharma? Or there's a lot of titles for orphan drug, can be ever for orphan. So actually there's an answer for that. So I'm going to set in the stage. So where are the biopharma industry now? And then what have been achieved? And are we ready for the next pharma? And in the legs, and so we will take a look for this first. Okay, so you know, everyone, why we were here. And then the biopharma business is growing just fantastic. So always, I don't want to say, because it is the double size, double rate, and for the increase, and also for the successful rate from the R&D. It's a long journey, you know, it's a long way to get into the marketing approval, no matter it's our pharmaceutical or it's biopharmaceuticals. But it's more than 10 years, it's getting there. But because of the technology and the development advanced us to allow us for the biologics, and it's growing just double size for that. So China, we want to say is right now it's becoming to the second largest the growth factor in the world for the pharma and the biopharma. You know, it's really hard to break down for the pharma and the biopharma, but we're looking at the molecular entity, we're looking at the molecular structures. So looking at the biologics and, you know, in the last few years and top selling, so the numbers of biologics is just keeping growing. So last year, it's 8 out of 10 the top sales are biologics. Not because I'm working in this field, but it is true. But you will see for that, you know, the Humira has been ranking the #1, the top selling in the 4th year, 4 or 5 years. In the next few years, and going to be that too. Okay, so we know the pharmaceutical is coming from the different raw material or different regions, but the biologics products are coming from the E. coli or the chill expression system. So right now, you know, the global regulatory agency, the growing rate and for the biologics even is being improved and even the faster. So for example, the FDA last year, you know, there's 13 biologics being approved and actually all are antibody-based, so out of the 59. So this is the fastest, the growth and for the approval. So right now it's more than 12,000 and the pipelines on the clinical phase globally. The next one probably is a little bit additional for you, but this is a collection for the top 10 sellings since 2015. So as you can see, the globally, the sales top selling, so the biologics and keep growing. Also, the Wall Street and the biotech performance also is double between the last 5 years. So in this aggregates, and the biotech-based performance also is the higher, is increased as time goes. So not just the patient like it, and the capital market. So this one is just came out like 10 days ago. It's for the top biotech companies and ranking. No matter it's true or not, because you just want to say because the China market, the Asian market, or Chinese market, and become to the second largest. So there are five of the Chinese companies, and is moved to or ranking to the top 25 biotech companies. Okay, because I would think most of the audience and are working for the pharmaceutical sector. So I still want to say what we are working and what we are looking at. For example, and the biologics antibody molecule, the molecular size is just so huge. It's compared to like, it's a aircraft. Compared to the small molecule, maybe it's like a small bicycle. So in terms of the complexity for the biopharma or the players, industry, how we get in there. So due to the molecular structure complexity, there are a lot of work to do. So we have seen the SFC for the small molecule for the prep purification, and also the one run for the purity and identity quantifications. But for the analytical, there's no single analytical methods can take us for the biopharmaceutical molecules, even for the identifications and the purity. So as you can see here, the biomolecules, not just only the size is so huge compared to the small conventional pharmaceutical drug. Structure complex. If we're looking at molecular-based structures, there are so much the post-translation modification. There's a lot of things, and from the basic molecular complexity or heterogeneity. So we always consider actually it is a mixture. It is never a single entity is present over there. So the stability, what you can see, we always know the biological drug, it is injectable. And it is the stability and needs the cold chain storage or transportations. So never can be at room temperature, the stability. So there is a requirement for that. And the modifications, many things and many ways, chemical way and enzymatic way. And so inside of that, the manufacturing and seems very, very complex. It's coming from all living cells as manufacturing facility. So even the production duration, it could be like could be 1 month, and compared to the small molecule, could be days. All right. So there's a single word. Let's say the characterization, structure characterization. In the small molecule world, there's no kind of that word. But for the biologics molecular structure, we're always using that because there's no single way and we can do and quantify 100%, like get all to know. So always there's a difficulty and to characterize. So we need orthogonal analytical methods to get a full understanding of the structure point of view. So mechanism for biomolecules. So many of us still, even it is a target, but still there's some downstream, the signal transduction is not fully understood. And then the drug delivery part, and the biological drug is all injected, as I said, and by the IV delivery or the sub-Q delivery. So there's no oral. So 10 years ago, we couldn't even think about any oral pills or capture for the biologics right now is become the reality, but it's working on the way. So there's safety issues, and even what we can see and for the pharmaceutical and compared to the biologics world. So there's immunogenicity issues. So there's occasionally there's some other allergic reaction. But however, for the small molecules, it's different, the toxicity and the safety issues. For the industry world, biomanufacturing, because I think today we're not talking about, the clinical studies part, but we're talking about, I'm coming from the CMC world. So first of all, looking at the global pharmaceutical, the manufacturing where we are. So it is very dynamic, evolving, and demanding. What we will see here, and everyone knows the WuXi Biologics, and as a CDMO, CMO, and how fast the growth, the business. So right now, the facility is worldwide, particularly in the Asian market, Asia facility location. So the products, and we make it, the orphan drugs, not permanent orphan. I will show you that. And for the biosimilar, generics, full-on biologics, and the cost of the goods significantly being reduced due to a lot of reasons. I will show you that as well. So the manufacturing facility right now is being very flexible. It's not just the fixed model, so it's modular. So anyway, single-use system and the efficiency of the production and the automation, what we talking about today, and also the monitoring in process control online, inline, PAT, QBD, and all the type, I would say it is ready. Okay. It is a crowded, this slide, but I want to emphasize it here, the CMC word, what analytical. The bottom is actually analytics and characterization. And so Shimadzu, like this kind of the vendors, and work for us. And they provide excellent breakthrough technology and for us to develop different type of the therapeutics and the drugs. So where for the biologics, the workflow, our process flow chart, the drug substance workflow from the living cell to the drug substance and the drug substance to the drug products used to be the regulation, in Chinese market used to be, has to be together, has to be facility feasible and not be separate. Right now, it's all involving. You do not have to be your own CGMP facility required. But even for our analytics, so this morning we listened to the L for the drug discovery and simulation. So analytics is being used for the early stage, the candidate discovery, developability assessment, and the pre-analytical, and make it feasible for the clinical studies. And then I move to the later stage of the clinical and ready for the launch commercializations. So different type of the analytics and the GXP quality systems allow all of this, the world right now, biopharma industry, and becomes to the de-risk management. So the risk is not that high anymore. So the QBD concept, initiative, and implementation is not just only for the pharmaceutical. Right now, for the biopharmaceuticals, it's very, very actively is being implemented. So the quality standards continuously improved. So the QBD and the process validation and according to the analytics, the synergy is always been there. So from the process early stage and from the quality standard QTPP and to the CQA to the process and the CPP and to final control strategy. So it's all being over where and for the biologics antibody drug development. Okay, so I guess this slide is a little bit extra because and for the biologics and everyone, and consider the analytics as like plain eyes, and also acting as ears, and can listen, can guide for the process development. It is a black box of the biologics process, and you cannot really to do that. There's a lot of things happen. However, analytics can tell you something. So analytics and for the biosimilar development, and we always can use that word, guide process development for the biosimilar. Okay, so everyone knows, and right now within the breakthrough analytic technology, state-of-the-art technology, can reduce some of the clinical studies, so it is more sensitive and make it feasible. So I will show you a couple of examples, and we get in there. Overall, it's due to the time constraint, and then I give you examples. And for example, the antibody drug, so the innovator and the sales entry, and how much the year cost or per milligram cost. So the year, the yellow column is here, as you can see. So from the drug substance, and that we estimate how much per milligram cost. And then if right now combine our integrated and the advanced the continuous manufacturing system, and we significantly reduce the cost. And look at see. And so with the medium, the first cost, the second large is the resin, and and then the third is the resource. So I would say, no matter it is very, very expensive antibody, and the patients not be able to afford right now, affordability is not easy. So even this one, for the orphan drug, the originator, it is orphan drug. You cannot say how much it is. So right now, we make it feasible. It's just very cheap. Of course, the orphan drug is not just defined by the cost of the production, but also by the population of the patients, But we were low, and in China, and the not so large population country, there's no even the orphan drug, the concept or the regulation that part, because there's from the population of the patients, it's not real orphan, and just from the number of the patients. So looking at this way, so within the advanced manufacturing technology and the integrated manufacturing continuous, and the conventional fed batch, making this, if we utilize the plant, the efficiency is getting higher, and then the cost is getting lower, and right now it's becoming to the continuous manufacturing, the cost is all reduced to $100 per gram. So this is really amazing. So combine, from the upstream to the title level, to the cell culturing, the duration, and over the downstream and over years, and then per batch, and how many lots per year, and the plant utilization. So due to the IP, so I'm not able to show my company, but this one is borrowed from the the company located in Hangzhou, China. So it's borrow their slides. Anyway, so I want to give you the first example. This is come from Biothera. Just 1 week ago, it's gained the first approval in Chinese market. So it is a Humira biosimilar. So how much we have done? Look at this, the analytics. So we have utilized 33 physicochemical methods and 20 bioassay methods deployed for demonstration from the comparability of the batch, different scale, process change compared to the originators from the different market, in Chinese market, US, and the EU. So all three arms comparison demonstrate from the analytical characterization and to the non-clinical and clinical pharmacology and to clinical outcome. So based on the stepwise approach and to totality of evidence. finally we gained the first approval. So some of the representative data, we have totally over 55 analytical methods. So just to show you the charge variance, the profiling by IEC, the glycosylation profiling. Okay, and then those the heterogeneity of the charge heterogeneity and also the glycosylation heterogeneity. And this is a high order structure by CD and DSC. And then the potency assay for the cell-based and the non-cell-based. So compared to the originator, the similarity, it is there. Okay, so Biothera is based in Guangzhou. We vision to go to the global. So the left side is our manufacturing park. And the current R&D is within the accelerator, but we are moving into the international bi-island, and this is Biothera. So we have more than 20 drugs and, in the pipeline. So 5 in the later stage clinical, and 4 of them is go to the global. So 6 in the early clinical stage. So we have more than 10 in the preclinical development. Another 2 examples, and very quickly go through, just based on my previous experience and in US Momenta Pharmaceuticals. So it's all based on the state-of-the-art analytics and to demonstrate how this molecule, the Lovalox by generics, so eloxaparin and molecules, we demonstrate that is based on the equivalency of the physicochemical properties, equivalency of the subunits, depolymerization, and equivalency of the biologics and biochemical assay, equivalency in vivo, without clinical studies. So this is really a made history in US, because for this type of the drug, and there is clinical studies required, but analytical allow this is made possible. Another case is carboxyl generics. It is polypeptides mixture. It's a huge mixture. How can we get in there without clinical studies? So the same thing, we apply integrated analytics technology and made the generics, and also being approved in United States 4 years ago. So this is another case. All right. We see the collaboration from the Shimadzu, and we have see all the collaboration, is very open. And so getting to the marketing approval, and we need to, based on the quantity of the science, the quantity of the data science, and the quantity of the science is based off the recognition. And always there's some work to do. So as I said again, and for the biologics, there's no single technique take us there. So we need orthogonal and multiple orthogonal methods. So like 3 cases I gave to you, and the other 2 cases. So we combine both of the physicochemical and bio assays. So usually around 60 methods have been deployed. So with the analytics and the calculation, and many of us are allowed to foresee the data of the quantity and the batch-to-batch the comparison. All right, so I need to wrap it up. So to go to the global, no matter where we are, and no matter what we deal with the molecules, so even the small molecule is the antibody, and the more next generation, more advanced molecule, bifunctional or ADC the type, small molecule conjugated to the antibody-like. And if we do the integrated CMC, and CMC is not just only the retest, the release test, and we need to synergize with the process from the preclinical, clinical early stage to the commercialization. So we can make, no matter it's a first in class, best in class, and a novel molecule. So finally, we'll make this globalization is feasible and combine with our manufacturing and the distributor. Thank you, thank you. I hope I have the answer for all of you. So next farmer, biopharma, it is the next farmer, and also the orphan drugs, no more orphan. Thank you.
Thank you, Chloe. So our next speaker is a professor at Kobe Pharmaceutical University. Please join me in welcoming Dr. Chikako Yamota.
I'm Chikako Yobota from Kobe University, Kobe Pharmaceutical University, but also I'm invited researcher in National Institute of Health Sciences. And the data today I shown was taken in NIHS. So my presentation is the regulatory science of generics in Japan. And that depends on the many analytical technologies. Yes. Yeah. Next. I would like to divide my presentation to 3 parts. At first I'll look at the current bioequivalence guidelines in Japan. At the present time, there are 4 guidelines for oral products. Guideline 1 describes the principle of the procedure of the bioequivalence studies of generic product to ensure the therapeutic equivalence of generic product to innovator products. And the fundamental way of the human test is almost the same to other regions. But the particular point of Japanese guidelines are they use many dissolution tests. That is much different from another regions. And number 2 describes the bioequivalence studies for post-approval changes in the component and the composition. Number 3 is the guideline for products which differ in strength. Number 4 is the guideline to change the dosage forms. And number 2, 3, 4 is also applied to the innovator products. This slide illustrates the pH and other conditions of various regions in the gastrointestinal tract. The pH in the stomach varies from 1 to 6.6, and that in the duodenum varies from 3 to 7. And intestinal from 5 to 6. The in vivo conditions are very dynamic and show the inter-subject variability. On the other hand, the in vitro dissolution test is simple and static. As you can see, for bioequivalence assessment, it is important to perform a dissolution test under multiple conditions by considering inter- and intra-subject variability, gastrointestinal conditions. In Japan, the Barty dissolution test plays an important role in the bioequivalence test. For bioequivalence assessment, it is important to perform a dissolution test under multiple dissolution pH conditions by considering inter- and intra-subject variability of gastrointestinal conditions. The dissolution profile obtained in all pH conditions are similar to each other. The majority of drug product is expected to be bioequivalent. Only a small minority is expected to be inequivalent. This table shows the dissolution test conditions to be investigated for each case of API. In the guideline for bioequivalence studies of generic product, multiple dissolution media such as 3 kinds of buffer solution and water are designated. In case of poorly soluble drugs, the addition of a surfactant was permitted. In Japanese guidelines, there are 2 ways to compare the dissolution profiles. There are also 2 criteria, similarity and equivalence. For example, when the average dilution from reference products reached 85% between 50 and 30 minutes in this case. One acceptance criteria of similarity is the average dilution amount of the test product does not deviate by more than 15% from that of difference product. At two time point, when the average dilution amount of the difference product is around 60% and 85%. And another way is to use F2 functions. This is the same way of another regions. The F2 value should be not less than 42. The comparison of dissolution is the particular way in Japan. Next, I move to the evaluation of generic product in Japan. We analyzed once the 36 tablet of ibuprofen in 1991. In Japan, the dissolution test was applied at first in 1995. So this is the ibuprofen tablet results. In Japan, the dissolution test was applied at first in 1995. There are no dissolution tests in JP in this time. So we used the test conditions of USP22, phosphate bifar, basket method, and not less than 60% in 30 minutes. You can see here in 36 commercial tablets showed various dissolution patterns. So we think this time all the products can keep sufficient quality without dissolution specifications. That is, we feel this is a big problem. So the big project, the evaluation of oral products, started in 1997. Objectives of this project is for all the products approved before 1995 to assure the quality of generic drugs by comparing dissolution profiles between innovator and generic products. And also to establish dissolution specifications for innovators and generics. One purpose is set the specific, and the other is the get the similar profiles between innovator and generic products. This slide shows the flow of the evaluation. At first, the dissolution profiles of the innovator drug info media were required, and the innovator company proposed the specifications. They make specifications for their product, and we check the specification and the method is okay, and then the generic product test following that method and compare to the standard products offered by the innovator companies. They compare to the standard product and they make their product to get similar profiles of dissolution. And if they cannot get the similar dissolution profiles, they can change their product and fit to the profiles to the innovators. Or sometimes they withdraw the approval. After the evaluation, the dissolution profiles of ibuprofen tablets changed to the right side. In 2008, this profile changed to the, and the similarity can be acquired to the innovator products. The evaluation of existing generic products finished in 2007, and for 10 years, it takes for 10 years. And all the products approved before 1995, 657 active ingredients and 4,133 products was checked, passed the evaluation, and finally 1,453 products withdrawn. And in this evaluation, we use this automatic dissolution test instrument. Almost Japanese dissolution instrument connected to the UV visual spectrometer supplied by Shimadzu. And this automatic dissolution testing instrument was given from the ministry. 11 testing institute was given this automatic dissolution testing. All these results in the evaluation were published as the Pharmacopoeia Codex, so by name Japanese Orange Book. And the Japanese Orange Book describes the dissolution specifications, dissolution profiles, media, the physicochemical properties of active ingredient, and the list of the evaluation products. This book is it contains many information of all the products. An example of dissolution profiles of a standard product for norfloxacin and for medias. This standard product was supplied by the innovator companies and distributed to the generic companies. And they compared the profiles by themselves, checking two products. And also, the structure and physicochemical properties are included in this book. And these results are included in the home page, and all profiles and characteristics and the list of evaluated products were shown in this home page. For example, we have only Japanese, but here we can see the dissolution profiles. Here, dissolution profiles and the properties and the final specifications we can see here. After the evaluation, for example, for loxoprofen sodium tablet, tablet 1 is innovator and other 23 is generic drugs. For 24 tablet, the profile is so narrow range. Very good result was obtained in this case. And next example is this amantadine hydrochloride tablet. We can result in specification test solutions, but in other solutions, we can find some differences of dissolution profiles. And the similarity of dissolution profiles in the specified test solution was achieved much better than the other test solutions, because people don't always perform the other solutions test. So this may be due to the failure in scale-up or others. So, for batch-to-batch consistency, depending on the concept of B guidelines, the dissolution test under multiple conditions where the dissolution profile under the discriminatory condition is preferable for the batch control. And the Japanese Generic Medicine Association has decided voluntarily to check the dissolution profiles periodically under multiple conditions for batch control. Specification is in release test is minimum requirement. The evaluation is finished, and next, we started the next activities. Recent activities to confirm the quality of generic are started. 2007, MHLW action program of promoting use of generics was started, and in that action program, for quality assurance, they write down that claims from medical providers are sometimes shown that generics, inferior quality, different efficacy, and safety. Some generics are different from innovator drug of the dissolution and over blood levels, etc. So governments want to make an effort, strengthen the on-site GMP inspection and sampling tests of commercial products by preferable laboratories and open their results. And also coordinate the expert meeting considering quality information, including medical providers' claim and scientific papers that show the concern to use generics. So that coordinated expert meeting is named Expert Committee of Generic Quality Information. MHLW requires the scientific and analytical support for confidence of the quality of generics. So this committee belongs to the NIHS, National Institute of Health Sciences, and the chair of the committee is the director of NIHS. And numbers of scientific reports concerning the quality, efficacy, or safety of some products increased. So MHLW and NIHS try to show the high quality of generics by testing the commercial products. The content of from 60 to 80 research articles and conference presentations regarding the quality and the clinical effort of generic drug products in Japan has been assessed by the committee members every year to determine their relevance. One example of unscientific report reviewed in the expert committee is shown here. After administration suspensions of one tablet orally to mice and compare onset time of sleep and duration of sleep for sleeping drugs, and the content of API were measured by HPLC, and sampling was made as follows. A tablet was powdered, and 2 milliliters of ethanol was added and sonicated for 30 minutes. This method is not the usual way described in pharmacopoeias, much different. So sometimes, at least in Japan, the researchers in universities or the pharmacists don't know how to check the release test. So they use their own way and show the mistake. So we sometimes the amount of one product gives 72%. So the decision of the committee, working group confirmed the amount of tablets and also checked efficacy by dissolution test. And we check. And there is no problems, and assay and acceptance value of uniformity of dosage forms, no problems in the 70 generic products are shown. And next, the dissolution profile is very good. This was evaluated already several years ago. So the good dissolution profile was taken for this, in this case. So we can show the high quality in this case. Next, The similar profile was obtained also for many other drugs. The 15 generic products, we can achieve a good resolution. And not only the resolution profiles, but also sometimes we perform an impurity test for preparations. One example is for cisplatin, for cancer drugs, and orange color is the innovator ones, and others is generic product. And the similar profile was obtained, and all is not so bad. We can confirm the quality of generic product by testing HPLC. We use in this case, we use the EP, Pharma Europa method, and JP is a little different. Column is used, and application is but the EP is best, we thought, so we show this result. Another example is this. Sometimes for cephazolin sodium injection, the doctor told us that sometimes the generics show the side effect, fever or others. So we analyzed the cephazolin sodium injections by LC mass IT-TOF. We tried once. And then I want to separate the impurities in test conditions published in JP once and want to analyze several peaks by the IT-TOF. So I told the Shimadzu members, I want to do so. So they offer the co-sense system before to LC mass IT-TOF. So this is very good, I think, because for mass condition is much different for pharmaceutical conditions, and we cannot confirm the peak, what is the peak in pharmacopoeias. So I use this for cephazolin sodium injections. Very good. This is a system, a little different shown in the Shimadzu website, I think. But here is a loop. The one separated by the condition pair, and the sample was, this is the pharmacopoeia's conditions, and sample was loops and condensed and sent to the LC mass IT-TOF. That we can condense the impurities, and also we can check the peak in IT-TOF mass. That's very good for us. Yeah. 1 minute. Oh, okay. Yosh. Okay. This is the result, and very good. And the genetic, all test results over 1,000 products are shown in the homepage of NHS. So please see this page, all shown. And recently, the Blue Book information package of quality of prescription drugs are shown as Blue Book and also NHS homepage. All results of the evaluation and scientific meeting and the government test result was summarized in Blue Book. So only Japanese, but please see this page. Many information are included in here. Thank you.
Thank you very much, Dr. Yamada. So next we have a speaker from India. Dr. Ravi Kravidi is the head of the analytical division at Syngene Research Center. Ravi?
Hi, good afternoon, everyone. I'm pretty excited to be here. Thanks, Shimadzu, for inviting me to this event. On behalf of Syngene, I thank everyone, the audience here, and also the organizers. And my special thanks to Spinco for helping me with the travel arrangements. And yeah, my talk is going to be more on the biopharmaceuticals. Of course, the topic has been Pharma Summit. As my former speaker has mentioned, there's a transition happening from small molecules to large molecules. In fact, I would probably in the future, the Pharma Summit is going to be a Biopharma Summit. So let's see how things move. Yeah, the title of my talk is Analytics for MABs, with focus on the biosimilarity assessments and stability studies. I've got a couple of case studies. And in fact, the word analytics is a little more inclined towards the software side. But I would say in the space of biopharma, it's more on the characterization part. As my former colleague speaker was mentioning that in terms of the characterization, especially in biopharmaceuticals, the large molecules, there's an extensive time that needs to be spent for the characterization of the molecule. And I come from Bangalore, which is the software capital of India. And so the word analytics is a bit borrowed from that field, I would say. And this is going to be the outline of my talk. I'm going to give you a little introduction about Syngene, the company that I represent, and give you a little more insight into a new laboratory, which is called CAPS, and highlight upon the services that we offer from the CAPS laboratory. And I'm going to focus on a couple of case studies on the LCMS analysis in terms of the peptide mapping, the intact mass characterization studies, and stability studies. And yeah, Syngene is a global CRO. We offer services to global clients, offering solutions to help them in solving the complex R&D problems. We are located in Bangalore. We are an integrated organization focusing on the drug discovery, development, and manufacturing services. Of course, we are also into small and large molecules. Large molecules I represent the large molecules division, the biologics of Syngene. And we are focused on monoclonal antibodies and therapeutic proteins. And of course, ADCs is another segment, as my former speaker was mentioning about ADCs, where the small molecule is coupled to a monoclonal antibody. So we have been into the field of antibody drug characterization studies. And in terms of the scientific ecosystem, Syngene campus is located in Bangalore, which is about 1.4 million square feet. And in terms of, I know in terms of WUSHI, we are not probably that big, but we are about 4,600 professionals. And we have state-of-the-art analytical equipment in terms of in the field of biologics. And in coming to the IP space, we have a pure play CRO with no internal programs or IP competition. And we also have these marquee clients where we specifically tailor-made our services to fit into their requirements. And of course, the quality focus is one of the major emphasis. The key areas of focus for Syngene, I would say, is safety and quality. These are the two key pillars for us to operate. And introduction to CAPS. CAPS stands for Center for Advanced Protein Studies. This is a state-of-the-art advanced national analytical facility located in the campus. And in fact, this is a very new entity, I would say. It has come up from the prestigious grant, which has come under the National Biopharma Mission. This is funded by BIRAC, the Department of Biotechnology unit. And as you've seen in the other slides and also in the Shimadzu videos, there has been a more focus on the academia and industry. In fact, in India, it's for the first time where you actually see a partnership between government and industry. In fact, this fosters the pace of drug development and product development in India. So this is a unique or first of its kind in India, I would say. Thanks to the funding agency, we have received close to about $4 million to establish a state-of-the-art analytical facility. And we offer end-to-end as well as modular analytical services. When I say end-to-end, it could be from gene to GMP, which is like the strength of the biologics unit in Syngene. At the same time, we also offer modular analytical services. In terms of characterization services, it could be intact mass analysis. It could be BIACOR or bioassay services. So those are the areas that we typically help our clients. And in terms of the technical services, we go through the entire spectrum from the method familiarization, where the client may have a method, so we help them, we tweak the method from the client, or we establish our own method and do the method development. And we also have a set of platform methods which have been working over years. And we do the method development and then the method qualification, followed by the transfer to the QC lab. This is the regular journey that we do in our laboratory every day. And this is a quick glimpses. You see here the secretary of DBT and our MD, Dr. Kiran Mazumdar. They've funded this new laboratory, and in fact, this laboratory, I would say, is one of the most expensive areas in the entire campus, considering the space in which we have placed all the high-end analytical equipment. And we are in the process of adding new equipment. Right now, what you see is one of the UPLCs next era, which is coupled to the mass spec. And in fact, CEMS, I would say, is one of the unique CRO combination in a CRO facility in India. And we are in the process of adding up more equipment. And here you see my colleagues working in the CAPS lab. And in terms of the infrastructure, here is a list. What you see, starting from the QTOF, from the different vendors. We've got a couple of QTOFs, and we've got a CE-coupled mass spec. We've got an image capillary electrophoresis, the conventional PA electrophoresis, capillary electrophoresis equipment, then the Biacore equipment, the analytical ultracentrifuge. All these are the set of analytical techniques or technologies that are indeed needed for in-depth product characterization studies. It could be biosimilars, or it could be any biotherapeutics. And whatever you're seeing on the screen is we are pretty much like a 6 months young group, I would say. Though biologics has been there since the last about close to a decade, but in the last 6 months, we have established all these new technologies in our lab. And adding further, as the former speaker was mentioning about the areas of biosimilar, the bioassay areas where she has shown the data on the Humira. So in fact, we do a lot of such services, not only for biosimilars, but also for therapeutic proteins, where we do extensive bioassay services. And we use different equipment which are needed, for example, the Biacore, or it could be the different the plate readers that we use for readout, the conventional ELISA-based techniques, etc. And here is the battery of tests, as you mentioned about. There are 33 battery of analytical tests that are needed for the product characterization. The product characterization can essentially be classified into 4 different categories: the primary structure, the secondary, the tertiary, and the quaternary. So for the primary, what we do is use the mass spec, which is intact mass analysis, and the peptide mapping studies. And for the oxidation of the variants and the deamination, we use a classical HPLC-based approach. We also use a HPLC-coupled mass spec to identify the site-specific modifications. And for aggregation, we use a combination of tools, starting with the size exclusion, and then the SECMOLs, which is a recent addition. We've been generating data on the SECMOLs. And analytical ultracentrifuge, this is another tool which is used for aggregation analysis. Similarly, the fragment analysis, glycovariants, disulfides. So these are extensive, in-depth characterization studies that are needed if a molecule has to graduate to the level of regulatory approvals. And for adding further to the secondary structure, it is the analysis by circular dichroism studies, and then the tertiary structure by the near UV and the fluorescence spectroscopy, et cetera, so on and so forth. And there are other studies in terms of the process-related impurities, which is the host cell proteins and the host cell DNA analysis. So these are our routine service offerings that we do from CAPS Lab. And focusing a little more on 2 specific case studies. One is on the peptide mapping. I'm not able to reveal the names because of the confidentiality aspects. So what you see on the map here is the digest of a complex protein, the panel on the top, sample A, and the sample B at the bottom, showing about a 95% sequence coverage on both the areas, inferring that there's a high degree of similarity at the peptide level between these 2 different innovator and the biosimilar components. And this is possible. We've done it on a HPLC separation. It's also possible to do it on a UPLC, cutting it down to about maybe 40 minutes or 35 minutes. Right now, it's about 110 minutes method. Similarly, here is another digest. In fact, I'm not doing a full justice to this particular slide when I say that this is a digest profile of 2 different proteins. In fact, we had spent more than a week to arrive to this set of data because we've used combination of enzymes. The extent of sequence coverage, what is expected, was about 95% in this case. But this particular, the amino acid sequence has more than about 50 amino acids, or the N-terminal itself had 50 amino acids without any specific residues which are accessible to any digestive enzymes. So we had to use a combination of enzymes to ensure that we get a complete sequence coverage. So right now what you see is a combination of lyse enzyme and asparaginase. So we've used 2 different enzymes to achieve a high sequence coverage of over 85%. And here is another analysis where we have done the intact mass analysis of rituximab. Here you see the charged envelope, and then the deconvoluted spectra on the other side. And similarly here, once we reduce, we get the light chain and the heavy chain information, which gives more insight into the extent of the glycosylation that's present on the molecule. We do this on a routine basis. We also subject the molecule to a PNGase enzyme, which is an enzyme to strip off the glycans. We do a treatment both PNGase with and without. So this is another example where you're seeing the PNGSF treated sample. What you see here is the intact mass, and here the one with post-deglycosylation. Similarly, these are 2 different batches that have come to us, intact A and B, and with and without glycosylation. And similarly, moving to another smaller protein, which is a smaller therapeutic molecule, romiplostim. Here you see the deconvoluted intact mass spectra, and then the deconvoluted onto the reduced mass spectra on the right side. And as I mentioned to you, the in-depth characterization studies do not limit only to the primary structure. We do a battery of tests, the 4 different categories of tests, the primary and the secondary structures. So circular dichroism is another analysis that we do to understand the secondary structure of the molecule of interest. Here, what you're seeing on the panel is the CD profile of romiplostim. And this is also a CD profile, the near UV CD analysis of romiplostim. And similarly for rituximab. I'll spend a couple of minutes on this before I close it. This is an exclusive study which is done on the mass spec to understand the stability of the immunoglobulin protein, the monoclonal antibody. The stability assessments are generally done on the HPLCs, but here we have used an innovative or a new technique to use a triple quad mass spec to identify and quantify the extent of modifications happening, which lead to the which can give an insight into the stability of the molecule. As you all know, triple quad mass spec is there's a targeted approach which helps in identifying the specific peptide of interest as compared to a global profiling approach. And for this, we use a targeted MRM or what you call as a PRM approach, which focuses on the specific analyte here. The specific analyte of interest is the peptide coming from the CDR domain of the monoclonal antibody. And what we have done in this case is we have subjected the protein, which is a monoclonal antibody, to forced oxidation process using different conditions like 10% AAPH or 0.075% hydrogen peroxide. Once we subject the protein to high oxidation conditions, so we asked a question, can we identify this using a mass spec? And once the protein or the monoclonal antibody is subjected to oxidation, there are specific sites on the CDR domain, which are the most unique segments of the monoclonal antibody, to identify those specific components. So what we have done is a tryptic digestion or a proteolytic digestion using lyse enzyme. We also worked on a combination of enzymes here. Right now, the data that I'm showing you here is a lyse chromatographic profile of the digest that has come from this monoclonal antibody. And we have done about 150-minute separation method. And further, what you see is we were able to identify specific peptide which leads to the oxidation conditions of this particular monoclonal antibody. So what you see here in the cartoon is the representation, what I was talking about, the triple quad mass spec, where it can focus on the specific analyte of interest amongst a complex mixture of analytes that get into the triple quad. And so what we have done, the cartoon here represents what we have done in terms of the workflow. So the question what we are asking is the oxidation of tryptophan residue in the CDR domain of the monoclonal antibody. For this, we have used the digest, as I mentioned to you in the earlier slide, the lyc digest, and then we've also spiked it with a synthetic standard containing the tryptophan residue. And it was analyzed in the triple quad. So what you see upon oxidation, there's a shift of about 16 Daltons. If it is a monooxidation, it will be 32 Daltons if it is a dioxidation. So what you see in the table here is a synthetic peptide, which is a standard control, and then our monoclonal antibody, which is not oxidized, as another control. And we have synthetic peptide, which is oxidized, which is used for spiking studies, and the forced oxidation map. So what you see here is the area which indicates the level of quantification, the extent of quantification, and the mono and the dioxidations that were identified using the mass spec. Well, these are the specific transitions that are used. I'll not go more into details. We use a software Skyline for generating the Q1 and Q3, which are basically used for telling the mass spec to identify these. Thank you for your attention.
Thank you, Ravi. So we're moving ahead now to Mr. Luciano Rosado. Luciano is the Quality Systems Director for Prati Donaduzzi Pharmaceuticals in Brazil. Please welcome Luciano.
Thank you. Is it everybody hear me? So thank you for Shimadzu to invite us to share our business case today. So I have some missions today. The first one is make you understand my accent. If I make you to understand my accent, 15% of my mission is done today. So I'd like to share some efficient-based approach of our company. So there are some cases we have a whole pipeline of efficiency in our company. So I'm going to share just a part of that. So it will be a brief presentation of that. So first of all, I'd like to show a little bit about the Brazilian market to you understand how does it works and some of our partnership with Shimadzu. So something about our mission, vision, values. Going deep in our mission that is provide health and well-being through high quality and reliable pharmaceutical products. So everything born from these three issues. Brazilian market is a great market because it's demanded for 210 million people, both private and public access for the drugs in Brazil. So it became one of the best market, on the great market at the world. So the whole market have the revenue of $24 billion a year. What means more than 230 billion doses sold in both markets. The specialist, my company is a specialist on generics. And in generics, we have $3 billion of selling by year. What means 56 billion doses made. And something about our competitors here. My company is the first one on generics in Brazil. So what means almost 11 billion doses made by year. What means almost 25% of market share on generics. And here you see our progression year by year. What means that design revenue for this year is $280 million of selling. So what I'm going to explore a little bit deep today is analytical technology. So we have 3 labs, including R&D and quality control labs. And we have 15 GCs, 20 UPLC, and 77 HPLC. We have different partners for such kind of equipment. And our partnership with Shimadzu is limited for HPLCs. What became us the biggest partner of Shimadzu in South America, and other 21 analytical equipment. So here I have just some photos of our labs. So how this have begun? The roadmap on efficiency is focused in business issues. We have 5 important business issues for our company. And these business issues start in quality, safety, production, costs, and efficiency. The whole pipeline of the job that I'm going to show you today is based on efficiency, KPIs, and strategy. This roadmap passed by evaluation of business process restrictions and evaluation of our business priorities and evaluation of QC lab restriction and tactical process. What means synchronicity between one step by another step, that some restrictions could stop the progression or the productivity inside the company. So the evaluation on the complexity, and at the end, management decisions that will drive the company to have more efficiency in each step or for keep growing. So the business case that I brought today to share with you is the movement on the HPLC lab, OEE on HPLC chromatographies, approach on analytical trends, OOS, and invalid analysis, incoming material management and sampling process, and shift transitions management, and improvement projects and other. It's important to mention here that our company is a great producer of generics. What means that are commodities in pharmaceutical market. So efficiency is so important as a driver in the business. So we produce during 6 days a week in 3 shifts. So the level of production and how the equipment are occupied in the lab and production is so high. So this first case here, I show you a spaghetti diagram. I don't know if it's clear for everybody, but this diagram shows the movement inside the lab. We have studied how the analysts were moving inside the lab. And each one of these red lines is one movement of this analyst. In this case here, we have followed this analyst that is responsible for preparation of standard solutions, mobile phases, and so on, during 1 hour for one analysis. And we concluded that for performing this 1 hour of job, these analysts have done 62 moves. What means 30% of the time of total time available for this analyst consumed by movement. So the diagnosis after this evaluation. So we got that the time wasting during test preparations, interference between preparation test and test phase. Because in this case, there are 2 people working together, one that prepare all the solutions, standards, and sample solution, and another one that is responsible to run in the equipment. Analytical retest needs and analysis loss, because the person, all the people are running, so on, and the quality is one of the drivers, but sometimes there are overpasses on this standard procedures. The cause in this case is absence or fault of analysis plan and organization. Another one is the lab layout and material organization isn't ideal for the analysis. So all the actions that we have made were based on this cause. And the results for this case was the reduction of 53% of the movement, what means 29 moves for this case. Another one is part of OE or HPOC analysis. For this case, we diagnose that we have delay on the lead time that's necessary to release. Doesn't matter if a raw material release or finished product release. The cause was a long time to start the further analysis, and shift transition is not efficient, and deficient on the communication inside the working cells. As I have mentioned, for HPOC cells, we have more than one person working together, and these people are responsible for do the transitions and do the communication with the people of the following shifts. And this was the case that deficient communication will stop this process and became unproductive. So the result on this case, we divided here the measure on productive time of the equipment, productive but during the setup, and analysis that is lost, and stopped equipment. For this diagnosis, we focus just in the time of the equipment that stopped. So this last data here, what was important that we have measured. For sure that we have other initiatives to improve ongoing and setup and non-productive ongoing time. But in this case, we focus in stopped equipment. So what means that we reduce it 17% of the time, the whole time available for the equipment. But if we consider that the stopped equipment got 43%, we increased almost 50% of the productivity that the equipment were stopped between one and another analysis, or between the transition of shifts. Right now, I'd like to share some situations that outside to the lab, but that support lab operations. In this case, we have a sampling process of raw material release process. So the diagnosis in this case was the reaction from the sampling cell were based just on demand needs, and low quantity of inside the lab to be tested. And this second situation could stop the availability to do campaigns of tests that save time inside the lab. And the cause was over-demand and under-demand situations, inefficient working flow in the sampling process, and a lot of change in the needs forecast. And the change in the needs forecast were caused because we had a lot of troubles between sampling cell and QC lab. The result in this case was the reduction in the line in 40% for raw material and 50% for packaging material total line, and the phase-in and increasing from sampling to inside the lab. So I'm going to show that will become more clear. So if you see the darker part was the batches that had to be sampled, and the lighter part was the batches that were inside the lab to be tested. So after the actions in the months of May and June, we could phase the material from the sampling cell to the lab. And in this case, we could increase so much that could provoke the situation that maximize the operation inside the lab, doing campaigns, saving time, saving resource, saving chromatography's resources, and so on. And like the same that happens for packaging material is much simpler to be analyzed, but we phase it, the material to be inside the lab, and time by time we could reduce it, the total line of batches to be analyzed. So the last three slides here, I'm going to just share some situations that during the observations, during the remarks that we have seen during this increasement process, increasement projects, we have realized that if we changed a little bit the way of working, we could produce much more without need of new resources, more people, and so on. The first one was the conventional way of working. What does it mean? That when some demand came from the lab, an analyst take this one to perform the whole analysis end-to-end. And we just realized if we divided this process for organization, preparation of material, preparation of solution, and so on, working in advance, for example, doing preparation of standard solutions and sample solution one shift before running this analysis inside the lab, we could save time without need of more people. So we changed it from conventional structure to a cell structure. Another one is a situation that analysts facing during some troubles. We just realized in this case that some analysts were expert to solving problem and running the analysis again, treat an invalid analysis and running an OS and so on. And in this case, we faced that there were analysts that were stopped for so long. Sometimes we got 1 week stopped for some situation that could not be solved quickly. So doesn't matter what's written here, but we have written some diagram putting in several places inside the lab that define a standardized way to solve any problem before running the second analysis. So in this way, it's very clear when which kind of data have to be revealed and which kind of document have to be have to be revealed by this analyst that's facing problem. And each people that have to be involved. For example, the supervisor, a more experienced analyst, and a specialist in such kind of analysis to treat this trouble situation in a more efficient way. So in this way, we could reduce significantly the time of treating any analytical problem inside the lab. And the last one, but not less important than the other, is improvement projects. So after running some projects that were managed by the director and the manager of the QC, we just realized that if we could write one single way to run new improvement projects, we could spread this kind of project for other people. For example, supervisor and experts inside the lab to continue improving the process inside the lab. In fact, I spoke so quick, so these are the situation and the business case that I'd like to share with you. And if somebody asked me, Luciano, what you had, what was the best result that you have gotten during this improvement project, this efficient project-based one? And I'd say, I could. Shimadzu won't like to hear that one that I'm going to say right now, but I could postpone a lot of investment inside the lab to the future for the next years, and we could reduce the number of the people that work inside the lab. My company, just a minute, my company is improving the profits in around 15% by year in the last 3 years, and I could reduce it in 20% our crew, postpone the investment to the future. This is the material that I'd like to share with you. I say thank you, everybody, and thank you for Shimadzu for the opportunity to share our experience.
Thank you very much, Luciano. That was an excellent talk. We are officially into the future, so we're going to try to go quickly through the last 2 talks. We have 2 15-minute, very brief presentations, and then we'll go to a break. So please see the speakers out in the hall after this. We won't have time for questions. So with that, we're going now to switch gears, and we're going to talk about medical cannabis. At first, we're going to get an update on the current situation on medical cannabis in Thailand. So please welcome Dr. Pakakrong Kwangkao.
สวัสดีค่ะ Good afternoon, everyone. Thank you for inviting me to share the current situation of medical cannabis in Thailand, which is a little bit complicated. But let's see how much I can make you understand and see the opportunity of medical cannabis product in Thailand and also at all over the world. So for the first slide, showing you the current situation at global level, national level, and health facility level. I think I'm very sure that you know that cannabis is gaining more attraction among the public, especially patients who have disease that do not respond well to the standard treatment. For example, cancer, dementia, Parkinson, Alzheimer's diseases. So, from current research, there are around more than 500 phytochemicals inside the cannabis. But there are 2 major commonly mentioned, which is THC and CBD. According to World Health Organization recommendation, they recommend to add THC, which has psychotic effect, but it also has the medical benefit. In Thailand now, we use cannabis preparation with high THC to improve quality of life of cancer patients. They recommend to add THC to the same level as morphine. For CBD, which has anti-inflammatory effect and also it has many medical benefits. The pure CBD or CBD preparation with small amount of THC like 0.2% should not be under international control. And hopefully that next year, this recommendation will be pushed to the meeting of INCB, International Committee that control the narcotic drug in UN. And if they approve, it means that CBD will be freely moved among the country. So for the Thailand, we just legalized medical cannabis in February this year. Actually, cannabis and also hemp are still under narcotic drug list. Anyone who wants to grow need to get approved from Thai FDA. But the current law open more to be used for medical purpose. But at the beginning, the first 5 years, in the law allow only government agency to grow and produce herbal product, cannabis product to the patient, only government agency who has mission on education, agriculture, and healthcare. So far, there are only 4 government organizations got license to cultivate and also produce the cannabis, including my hospital. For medical cannabis product, due to insufficient scientific evidence, both on safety and efficacy, so there is no any product registered with Thai FDA. However, due to, social movement in Thailand, you may know that the government allow the medical cannabis to be used as an unapproved drug. What does it mean? It means that this product should be used only in approved health facility under what they call special access scheme. It means that when the patient go to the approved health facility, the hospital or health facility should have the protocol to monitor safety and efficacy and report all the results to Thai FDA. How about that at the health facility in my hospital? Since we are no model in developing herbal product, we are requested from government to develop the model to share the experience with the public, starting from raw material management. We do cultivation with a group of farmers, and we do extraction and production, and also we use the product, the medical cannabis product with our patient. The product produced by our hospital also distributed to the other hospital under Ministry of Public Health. So what we are doing now in Thailand, we collect, we breed, and improve the genetic of Thai native strain. You may know that cannabis is our local plant. We have traditional recipe, Thai traditional medicine recipe containing cannabis, which is recorded in our classical text for more than 300 years. And so far, the government announced that they allowed to use 16 recipes of traditional medicine for our patient. And we also developed the extraction technique. So far, there are 2 main techniques we use for extract cannabis in Thailand: alcoholic extract and SFE. And the third one is develop the cannabis medicinal product. Currently, there are 2 types of medical cannabis product in Thailand. The first one for modern medicine. There is only one preparation, oral oil drop. And the second one is traditional medicine, which is in the powder form. When the patient use, they mix with honey, regarding to our own knowledge. However, there are some universities developing the new dosage form of the cannabis, for example, patch or even inhaler, but they are all in the research and development process. And the last one is develop the quality control method of raw material and finished product. Now, Department of Medical Science, they are working with their partner to develop the quality control method. We try to control not only THC and CBD, but the other active ingredient like CBG, CBN, and also control the fingerprint and do the DNA fingerprint as well. And for the cultivation system, there is a big debate because in Thailand we open opportunity for farmers to grow cannabis. So there are a lot of farmers interested. They want to grow. They prefer to go in open air because they don't have to pay much money. But in terms of return, we don't know yet. So by next year, in my hospital, we have 3 systems. We have open, we have greenhouse like this, and we have closed system. Yes, sure, closed system is most expensive one. But we promise to the society that by the next year, we will know which one is worse in terms of investment and return. And for the production, for the manufacturing, the law require GMP standard for production. So any producer who want to get the license, they need to get the GMP standard first. And this is a production process. Right now in my hospital, we haven't harvest the cannabis plant yet because it's very urgent. You may know the law effective in February, and after that there are public movement. They are forced by our patient. They want to use the medical cannabis product. So the Ministry of Health organized several meetings, try to solve how to have the qualified cannabis medical product. So finally, we come to the conclusion that why don't we use the confiscated cannabis? But before do that, we need to check the quality. So Department of Medical Science, they check all the confiscated cannabis in the warehouse, and some of them, they pass the standard. So we use this one to produce the herbal product, the cannabis product, so sorry. And this is a specification. Yesterday, I just have meeting with Thai FDA and Department of Medical Science. By next year, in March, we're gonna have the national specification of raw material extract and also the finished product published in Thai Herbal Pharmacopoeia. But this one is specification that we use for our hospital. And for clinical practice, even we open for patient, but not every diseases that can get the medical cannabis. So far, we have only 39 hospitals under Ministry of Public Health that open specialized medical clinic. For modern medicine, I mean oral oil drop that we use, there are, and the data at November the 5th, there are around 1,882 patients who came to the clinic. But not all of them, just 1/3, around 600 patients getting medical cannabis due to limitation of indication, and also we need to screen many diseases. And this is for Thai traditional medicine recipe. I'm so sorry I didn't translate because the recipe is in traditional, in the old language. But there are 2, so far there are 2 traditional medical cannabis recipe. The first one used for insomnia and another one used for stroke. And so far there are around 18 hospitals provide traditional cannabis recipe. So to have you see more, to give you see more the opportunity of medical cannabis product, I will talk a little bit about the treatment result in my hospital. So in my hospital, we provide 2 times a month, to provide medical cannabis product to the patient 2 times a month. We have screening, interview, medical history, prescribing, counseling, and monitoring and evaluation. So we know that even cannabis has medical benefit, but they have a lot of side effects. So we need to screen and exclude this kind of condition. And this is a monitoring and evaluation program. The first dose the patient get, the pharmacist will call to the patient to make sure that they are safe and monitor effectiveness with many medical scale like PPS for cancer patient, PSQI for the one who cannot sleep well and fail the standard treatment, safety and compliance. And so far we have 3 types of cannabis preparation. The first one, cannabis extract with high cannabinoid, CBD and THC, and also the traditional recipe. So far for oral dose, we have 92 patients receiving. This is the result of modern medicine. The quality of life is improved around 70%. And this the treatment that we give, there are only 5 diseases. It seems to work well. I mean, around 70% of patients respond well and few side effects we found mild and moderate. The moderate, some patient who has kidney disorder, some of them return to the normal state. And there are 4 patients received the traditional recipe. Just only 4 of them, but all of them improve in terms of, we measure by PSQI. And this is the presentation, and I hope that you will see the opportunity of medical cannabis product. Thank you very much.
Thank you, Dr. Pakakrong. So it's my pleasure now to introduce the final speaker of this session. This speaker is the CEO of Front Range Biosciences. He's not only an expert in cannabis, he's also an expert in agriculture and clinical diagnostics. So again, it's my pleasure to welcome Dr. John Vaught.
All right, thank you, Scott. So I'm going to try to keep this quick because I know everybody's anxious to get some coffee and take their afternoon break. But I think you'll want to stick around for a few minutes because I've got some interesting things to tell you about here. So first, I just want to say a special thanks to Shimadzu for putting this together. It's been a great day, lots of good talks, and I really appreciate the opportunity to be here. And it really appreciate the hospitality shown by the entire Shimadzu team. So it's been a great time. If somebody would have asked me 19 years ago when I started my career as a young synthetic organic chemist working on new technologies for personalized medicine, would I be here talking to a bunch of pharmaceutical companies about medical cannabis, I would have laughed. But the world's changing, and it's changing very, very quickly. And the reason is because our understanding of this pretty interesting, extremely unique plant is advancing beyond what we ever had before. And it's no longer a plant that needs to be feared, stigmatized, or even prohibited. But based on what we know now, this is clearly a plant that should be researched heavily and, more importantly, exploited to improve human health and also treat disease. So really quick, I'm going to give an overview of just the history of cannabis and how we got here, a little bit about the chemistry behind it, some pharmacology, a little bit about the market opportunities and the current state of the industry, some recent scientific developments coming out of Israel in the clinical space, and then we'll wrap up. So before I started Front Range Biosciences, about 5 years ago, I spent about 15 years developing molecular diagnostic technologies in the human space, mostly leveraging novel biomarker discovery platforms to support the drug development, drug discovery process through the clinic. I got really interested in agriculture along the way, and cannabis really represented a very unique scientific opportunity as well as a business opportunity to bridge these two worlds of agriculture and medicine. Front Range Biosciences is headquartered in the United States. We're an agricultural biotechnology company, and we focus on cannabis agriculture, but we also work in coffee, which is a topic for another day. But we specialize in the research and development of new plant varieties. So new varieties of cannabis that have unique properties. And we have a pretty large R&D team with analytical chemistry, bioinformatics, genomics. We do a lot of advanced molecular-based breeding, and the whole point of that is to develop new cannabis-derived ingredients. As I mentioned, we're headquartered in the US, and we also have an outpost in Spain right now through our partnership with the CRAG in Barcelona. So just a brief timeline here. Cannabis has been around for well over 10,000 years, and it's been reported at least 10,000 years ago, probably originating not so far from this part of the world, in China, where it was used for textiles, as well as some medicine early on. And pretty much throughout the next 10,000 years, there was multiple examples where it was used, anecdotally as medicine. And up until about mid-1800s in the United States, when it reached that part of the world, where it was actually marketed and sold as a pharmaceutical for almost 100 years, up until about early 1930s, 1940s, when it started to be prohibited. Anyways, that eventually led to worldwide prohibition due to its psychoactive effects. And so our research has been very limited. But the last 75 years, things really started to change. In the late 60s, we had the discovery of the THC molecule in Israel. And then about 20 years later, in the late 80s, we discovered the cannabinoid receptors, CB1 and CB2, in the endocannabinoid system. Also, the first cannabis or cannabinoid drug was approved in the 80s. And then throughout the late 90s and into the 2000s in the United States, we saw cannabis legalization happen, mostly for medical purposes, although now we've seen recreational as well. And most recently in 2018, the first FDA-approved drug for seizure disorder in pediatric populations, Epidiolex, was approved. So we've really come a long ways, especially in the last 20 years. Today, it's now become a global industry, and there's some form of medical cannabis regulation in well over 30 countries worldwide, and there's also a couple with Canada leading the charge on the recreational side. So why is cannabis so interesting, and why should you all care about it? Well, thanks to companies like Shimadzu, and they've honestly been a pioneer in the analytical chemistry instrumentation space as it relates to the cannabis industry. We now know that there's well over 500 active compounds in this plant, which is pretty remarkable. Many of these are cannabinoids, so there's well over 140. I've listed some of the well-known ones here. There's a bunch of terpenes. It's also really interesting from a phytonutrient perspective. So not just medicine, but there's also nutritional properties here. The grain from the plant is a great source of plant-based protein, dietary fiber. It's got a balanced ratio of omega-6 to omega-3 fatty acids, just like the human body. So really interesting from a lot of different angles. It also can be used, the fiber can be used for industrial applications, textiles, construction materials. So really, really interesting crop. These are some of the most well-known cannabinoids, and I've shown these as the decarboxylated form. They generally exist as an acid in the plant. But what you see here is a lot of similarity between these and delta-9 THC, which is the most famous one that gets everybody high and has psychoactive effects. You can see how similar it is, but most of these other ones are not psychoactive. In fact, most of them are considered not psychoactive at all, and they have a lot of other interesting physiologic effects, though. Note down here, THCV, which is just different by a 2-carbon chain from delta-9 THC, and this one has drastically different effects than delta-9 THC. So we're really just getting to the tip of the iceberg here in our understanding of how cannabinoids are modulated or modulate the endocannabinoid system in the body. So in the plant, there's several different cannabinoid pathways. They all start with farnesyl diphosphate generally as a precursor. So they go through a series of different enzymatic transformations that leads to things like terpenes, different types of terpenes, and then some of the cannabinoids. This is a very simplistic view of the secondary metabolites in this plant. But the point here is that it creates an incredible amount of chemical diversity from just a few precursors. And if you were to ask a synthetic organic chemist, they can certainly make the same type of chemical diversity, but it might take dozens of discrete chemical transformations to generate this kind of diversity, and especially when you consider things like stereochemistry and the structural diversity that we see in cannabis. On the safety side, cannabis actually turns out to be one of the safest substances that you could consume. And there's tons of data to back this up, even recently in the Epidiolex study, which I'll talk about in a second. But the therapeutic ratio, this is a fun graph to look at, which shows basically the effective dose over the lethal dose. So it gives you a sense of how much you have to consume of a certain substance before you could have a lethal event. Well, look up here for a second. Aspirin, caffeine, yes, there's things like heroin and cocaine, which can certainly be deadly, but aspirin and caffeine have therapeutic indexes that are over 1,000 times more than cannabis. So nobody's actually ever died from an overdose of consuming too much cannabis, which is pretty interesting. The worst thing I think that has ever happened is people have panic attacks and they eat too many potato chips and then fall asleep for a little while. So in Epidiolex, it was well tolerated in a pretty sick group of children. So it was tolerated at 10 mg per kg up to 20 mg per kg, which in a normal adult represents well over 1,000 milligrams per day. Only 2.7% of the trial participants in one group, discontinued treatment due to adverse events. And those adverse events were actually, on the grand scheme of things, were not terrible. It was things like mild liver toxicity, kidney toxicity, and sleepiness. This is a fun slide just to show some of the main well-known cannabinoids and the different pathways and the different areas that they're implicated in. Things like anti-inflammatory, antipsychotic. There's a pretty broad range of applications for just these compounds. And as I mentioned, there's 140 of these. The endocannabinoid system we've learned a lot about in the last 30 years, and it's got the CB1 and CB2 receptors, which are present throughout the central nervous system, peripheral nervous system, so it's present in almost every tissue in the body. It's very important in key regulatory mechanisms that affect things like mood and pain and learning and memory, so it's implicated in a pretty broad spectrum of human disease and human health. This just gives a quick example of what I'm talking about. So the CB1 receptor, this shows two different modalities of how ligands interact with this receptor. Up top, this is endogenously produced ligands. So there actually are endogenous cannabinoids. They're not just produced in the plant, but our body produces them as well. And this just shows how it modulates CAMP activity in synapses. This one shows down here a different mechanism where it's involved in the beta-arrestin pathway. But the point here is really that these cannabinoid receptors are widespread throughout the body, and they're implicated in a lot of different physiologic pathways. The other interesting thing about cannabis that's been noted is this entourage effect. So the traditional dogma of one ligand, one receptor that is part of developing drugs in the FDA approval process. Cannabis actually is been observed to have very different effects when you administer groups of cannabinoids and terpenes together versus individual isolated or purified cannabinoids. So this is just a study showing the difference or some comparisons between aspirin, tramadol, some anti-inflammatories with pure CBD, and then an extract of a cannabis plant with a cocktail of different cannabinoids and terpenes present. And what they saw was not only did the cannabinoids have similar effects in terms of anti-inflammatory effects, but the difference between pure CBD alone and the combination from the plant extract was actually significant. So when we look at market opportunities, there's the process that we're all very familiar with here, which is going through FDA approval and the clinical process. The challenge, and a few other folks spoke about this today, is that it's not cheap. It can easily cost close to $3 billion today, and it takes at least a decade typically, unless you get some type of interesting status for an orphan drug or something. So there's several drugs on the market, but there's only a handful that are cannabinoid-based. And what you'll note is that all of these are some combination of THC and CBD, which are the two most well-known cannabinoids, as I've mentioned. So once again, there's 140 at least additional cannabinoids, and the only drugs out there today are basically derived from the two that we know the most about. There's also the consumer wellness product side. So this is really interesting space for pharmaceutical companies. They're well positioned because of their infrastructure, expertise, the ability to manufacture formulations, get through approval process. But this is much less regulated. And certain pharma companies have demonstrated a lot of traction in the consumer wellness space. And there's a huge opportunity with cannabis here, especially in the early days, mainly because it's less regulated, but also because of this entourage effect, so this multi-target, multi-ligand effect. So you can extract these reagents or ingredients as a botanical from the plant and get them to market fairly quickly in the form of dietary supplements, food additives, and then even superfoods. So I'm going to finish up the talk with just a few incredible developments that have come out of Israel over the last few years, just to talk about why this is actually interesting. So this first is just another example of basically of the entourage effect, and it shows that over here, so on this graph here, we've got a series of different plant extracts. So each of these represents a unique cannabis plant. Over here, we've got the percentage of cells of a specific melanoma cell line, and here we have the dose response. And what you see is that not all of these different cannabis extracts have the same dose response. Some show up at 4 micrograms per mil, some barely show up at all. And over here, if you compare that with this one cell line, A375, to pure THC, it has no effect. So it's not the THC that's actually killing these cells. This just shows some pictures of fluorescently stained cells and the cell death. And what they also found out was not only did certain strains or certain plant varieties affect the cell lines differently, but different cell lines had different responses. So it wasn't a broad effect against all cell lines. It was a very specific against certain types of cancer cell lines. Another interesting result has been around autism. And it's a developmental disorder in children. And there was a small study that was going on in Israel where they were looking at CBD and cannabinoids for autism. And they had to switch formulations during the middle of the trial. They were using a plant extract, and they ran out of one, and they had to switch to another. Well, it turns out it was a different plant. And what they ended up seeing was a bunch of adverse events. So they started to see anger, and actually one child even attacked his mom, so they had to stop the study. When they changed the plant. So they said, well, why is this happening? So they went in and looked at the two different formulations, 266, 267 here is how they're noted. And what you see here is different cannabinoids that are present, and these were all analyzed through generally chromatography. And what you see is that they have different amounts of some of these different cannabinoids. And some of them are pretty remarkably different, hence this kind of checkerboard pattern versus a series of stripes. So once again, demonstrating that there's a lot more going on here than just a few cannabinoids or one single cannabinoid that has these effects. So this is the last piece, which really demonstrates the chemical diversity. And I apologize, it's a little bit blurry here, but the point is these are examples of mass spec experiments and some tandem mass spec experiments that show cannabinoids over here, terpenes over here, and on the x-axis, these represent different plant varieties or different plant types, and then down here you see different cannabinoids, and then you see the quantitative amount of these different, or relative amounts, I should say, of these different cannabinoids across different plant lines. And what you see here is a huge amount of diversity, right? And most of these cannabinoids, we barely know anything about. They're produced at very, very low levels in the plant. They've barely been isolated. Some of them even haven't been synthesized outside of the plant. And there's an incredible amount of opportunity here to exploit this diversity. So, to close, clearly, cannabis has implications, broad implications, in human wellness and disease. It's a very safe class of compounds, and it can come from botanicals. So straight from the plant, which is a very cheap, low-cost way to extract these materials in the current state of things. And we're also going to start to see things or applications of synthetic biology. But I will mention that this is a challenging space for cannabinoids. There are a class of compounds that are generally toxic to microbes, like yeast and other things that you might produce these compounds in through synthetic biology. But last but not least, these compounds are going to show up as ingredients in therapeutics, wellness products, and even nutrition. So it's really an exciting time to be working in this space. And if you guys haven't done any research, I'd encourage you to take a look at it. There's some really incredible opportunities here. So with that, I'll conclude, and we can go get some coffee.
So thank you, Dr. Vaught. And now we're going to take a break, but please, since we're behind in the schedule, we'll just take a quick restroom break, grab some coffee, and please bring the coffee back into this room. So just like a 5-minute break. Thank you.
Okay, thank you very much for really taking the very, very short coffee break. And I'm sure you will not regret this cutting short the short break, because you have a better evening and longer enjoying time at Sea Aquarium. Okay? So please come back so we can finish in time and we can enjoy more. All right. So in the last session, or we can call it a grand finale, we have a panel discussion, and our panelists are already ready on the stage, as you can see. The first one from the left is Ms. Aristiana Permata Adji, Quality Assurance Head, PT Kalbe Pharma, Indonesia. And Dr. Thomas Pieber, Chief Scientific Officer, CBMed, Austria. Dr. Paul Heng, Associate Professor, Department of Pharmacy, National University of Singapore. Mr. Boru Che, Co-owner and Managing Director, Labina BIA Separation, Slovenia. And our own Maruyama-san, General Manager, Analytical and Measuring Instruments Division, Shimadzu Corporation, Japan. The moderator is already known, Dr. Satish Lele from Frost & Sullivan. So may I request Dr. Lele to start the panel discussion, please?
Okay, thank you, Sandy. And good afternoon and welcome to the final session for today. I know this is going to be much shorter than we had thought, but what would be interesting to is what kind of insights that we can quickly gain from, the whole panelists that we have here with so much of experience behind them. So before we start, let me quickly summarize about about the topic or theme for this particular discussion. So we are talking about the transformation of the pharmaceutical industry, how it is going to be disrupted, and what are some of the key aspects which are important and transformative over the next few years. Now, in Frost & Sullivan, when we were looking at the whole transformation of the pharma industry, we compared it between the 3 decades. So when you look at the past decade, it was all about blockbuster drugs and how you could commercialize them in a big way, and we have seen that happen in the past decade. If you look at this decade, it moved towards more of specialty and how you could add more value rather than volume. And when we look into the future, we see that moving into what we call it medicine as a service. Now, how that will evolve, I think probably we'll get some insight from this panel over here. So let me start with my first question to Tia. You have been in the pharmaceutical industry for a long time.
Yes.
And essentially looking at Indonesia and the Southeast Asia region. It would be interesting to know from your perspective, how do you see the future of the pharma industry for this region? What are the key issues and challenges that you foresee? And I mean, if you can give maybe 2-3 insights into how you see this transformation happening. I know the future of pharma is a very, very broad topic, but I think the insights mainly from this region and how do you see the future would be very interesting for the audience.
Okay, thank you, Satish. First of all, because I'm coming from pharmaceutical industry, so if we look to as the pharmaceutical industry, then we have to see that it is also responsive to the pharma for the manufacturing 4.0. Whereas the future of manufacturing has to be agile and flexible and also fully technology involvement here. So this session, this event, absolutely that it is supported for me to see and to make a broader perspective, which is also supported to reach the future of manufacturing. And then another things that I would like to inform that as the manufacture industry, especially in Indonesia, I'm coming from Indonesia, our company is also have a vision and a mission to be playing in the global player. So therefore, requirement, it has to be, we have to be explore and fully implemented to the technology, which is the PQS, as I think all of you are familiar with that we have to be strengthened on that. So related to the technology, so there will be, of course, many skilled people to be upgraded because they have to be validated, etc. So that is my perspective. Thank you.
Okay. Thank you. I think resonates very well because when I look at all of Southeast Asia, almost every country has their 4.0 program. You go to Malaysia, there's Malaysia 4.0, Thailand 4.0, Indonesia 4.0. And a lot of that digitalization will also impact the pharmaceutical industry as well. So it is quite important for the pharma industry to prepare to be ready for this whole digital transformation that is going to happen. So thanks for that. What is interesting is that we have the entire value chain of the pharmaceutical industry on the stage today. From the pharma industry, we have the CRO, then the equipment and the analytical suppliers to the pharma industry. We have the clinical research and the academia. So you can't have a more diverse team on stage. So it'll be very interesting to hear each perspective in terms of how they look at pharma, the future of pharma, and the kind of role that they would probably play in that development. So let me move to Dr. Bonut. Now you come from the CRO and also you're based out of Europe. I think it would be good to hear your perspective on the future of pharma from your perspective, and what would be some of the key insights that you would like to share.
Okay, thank you. First, good afternoon, everyone. So our CRO does quite a lot of work for big pharma, biopharma, for mainly US market and Western European market. We are GMP and FDA approved as well. In terms of pharma segments, which I think will be the future of pharma, is definitely biopharma and biosimilars, which we heard today also. One very interesting field where we see a lot of movement and inquiries are genetic therapies with AAV, so adeno-associated viruses, and where we've done a lot of analytical research also. And also, of course, a smaller market, but also there's a lot of discussion in Europe currently, also with legislation, is a cannabis market. It's now small, but it has one of the largest potential to grow, about 50 to 60% until 28. In terms of industry, of course, also in Europe, pharma, also small, big pharma, it's getting prepared for 4.0 industrialization and digitalization. But the biggest, let's say, the biggest focus of the talks, when we have, for example, an FDA inspection or where somebody from our partners comes to inspect us, is basically data integrity. So how to ensure that from the API to production to analytics, you have a fully compliant data integrity process that can be also followed from the beginning to the end. So this is what we see. This is the biggest also the amount of time that inspections spend in pharma or with CROs. It's about 2/3 is about data integrity. So that's one very hot topic at the moment in Europe.
Yeah, thanks for your insight. Let's move to the analytical. The analytical intelligence person on the floor. So it would be interesting to hear from Shimadzu's point of view how you foresee Pharma 4.0 and what kind of role companies like you are going to play as far as the whole development journey is concerned across the entire spectrum of the pharma industry.
Okay. From my perspective, when I listen to the speakers is saying, especially in the morning session, there are lots of effort on taking the cutting-edge technologies in AI, IoT, and that's all the new things to improve their productivity, efficiency. But those are the things they're looking at, and we have been trying to support all the pharmaceutical company by providing the efficiency, improvement in instrumentation. For example, ease of use is a kind of the value we can add on the instrumentation. And with the ease of use, of course, the operator can make more free time to think about the more variable things, of course. And at the same time, I think the lab managers can reduce the amount of the time for the training, for the new instrument, for example. But that will reduce the cost for the analysis, I think. And also, we can provide the deduction of the downtime, for example, by using the IoT technologies. We can provide the instrument situation, operating situation, and the service information to the service personnel through the IoT technologies, and they can make instrument operative always without the downtime. But this is one of the value we can add as well. And also, by using the analytical intelligence, we also can provide the customers, a pharmaceutical company, for the reducing the risk of the human error. Okay? For example, we have the mobile phase amount sensors in our new HPLC system, which send the amount of the mobile phase before they start the analysis. So if the mobile phase is not enough to execute all the batch sequence for the analysis, then before starting, the system itself will give a warning to the operator so that they cannot make a mistake in the analysis sequence. But that's all of the things is what we can do, and we can accelerate the effort of the pharmaceutical company to make it more efficient, I think.
Thanks. I see you have a very practical approach along with technology, because I think that's equally important, specifically for the people who are working there, to ensure that life is easier for them, specifically on the manufacturing side. I'll ask you a different question, Dr. Thomas. I mean, we have kind of talked about what is going to be the future as far as the pharma industry is concerned. But I would also like to get a sense of what are some of the important challenges that you believe are equally important to understand, and how are we going to overcome some of those challenges, specifically for the pharma sector?
Yeah, thank you. And thanks for being able to discuss this. Well, I think we all remember when the first human genome was sequenced, we had the promise in the room that with understanding the human genome, we might have answers to many diseases we cannot treat yet. And when you look how much has been delivered so far with this personalized medicine, we still have to deliver there. There's still a huge need. And the learning of the last 2 decades after sequencing the human genome, one main lesson was biology is more complex. We need more methods to understand. And we are now in the age of multi-omics. So sequencing the gene is one thing, but understanding how these genes are translated and what they do then in patients is where we stand right now. So I think we have to take in the challenge of multi-omics, and that needs analytical intelligence. I'm on the side of the people from today who said that with this huge amount of data, we need artificial intelligence, analytical intelligence to tackle the problem. The other thing I want to mention is that we should not forget many diseases that are neglected, orphan diseases or less common diseases. So pharmaceutical industry was focusing a lot on the big fields like cardiovascular, cancer, but there is room in between where the industry has to develop further.
Very valid points. So the final comment from the academician, when you look at all these issues of where the industry is heading, some of the challenges that you hear, I think from an academic standpoint, it would be interesting to know how you are supporting this whole transformation, as well as there were so many questions about skills, which Tia also mentioned. So how is the academic world preparing for the future of Pharmaceutical 4.0?
Okay, thank you very much. Of course, as an academician, we are well connected with the industry. But being in pharmacy school, our major job is actually to supply pharmacists in the hospital and the retail. And actually, the industry is actually a small part of the total picture. But however, I am working with the industry and manufacturing industry, so my focus in research is in the industrial pharmacy. Now, of course, when we are thinking of training manpower, we always start with this, that we have to think global and act local. Because ultimately, Indonesia will be very different from Singapore. Although I work very closely with Kalbe, but in training the worker. But in Singapore, you will look at the landscape. We do not have that many generic pharmaceuticals, but we have a lot of MNCs. So MNCs are here basically very different from, because they don't do basic work. They are just mainly a supply chain operation. So it's a bit different. But looking at the pharmaceutical industry in totality, I think there are 2 key activities that I think is very important. One is that in R&D or production, the QBD concept is very important, quality by design. Because here is where, gone are the days we just try out and see what we get. We have to think about how we are going to go about doing it. So quality by design to me is very, very important. The other is, you have now better and better machine. They do sampling faster, they measure faster, they get more and more data. But at the end of the day, all this data that is generated, what are you going to do with it? We need to think about data analytics. Of course, this is a very big field. How do we make use of all this data? Or we just think about the storage and the retrieval and the usage of the data. And of course, AI is important, but it's not so easy to put AI into the factory. But nevertheless, there must be some skill set that require the analytics to pull out the right data, to put it in a form that is more usable, more friendly. And I think this is something that is a bit of a miss. Although I do a lot of training in my students in QBD, PAT, and so on, but we try to get into data analytics, but it's not so straightforward. It's a very complicated. We work with Syntron and a few other companies, but it is not an easy process. But nevertheless, I think Singapore has put into a big landmark on AI. Hopefully, there are more ability to access data, to manage data, and to retrieve data, so that whatever we do, besides pass and fail, we can see beyond that what we are getting. I think that is what I will put up.
Yeah, good points. I think data, everybody agrees that it is the money of the future, but very few people have really understood how to convert that into something which is usable. I know we have very less time. I will just end with a final question to all the panelists. As you went through the whole discussion of today, listening to all the different presentations, can you share, all of you, one important takeaway from today that you would like to share, which was probably the most important from your perspective, and you can share it with our audience here?
Okay, if we looked into what presentations the whole day, this met with my expectations, because we are also going to improve our system. Actually, we are in the preparations of in the laboratory smart. So with today, in my opinions, I can see the real example, the best practice, which is actually like the real life. So that's from my point of view.
Thanks.
Well, all presentations today had one thing in common. Without analytical instruments, there would be no presentations. No summit in Singapore, so everybody, you will not hear.
Shimadzu should be very happy with this.
No, but that's the truth. So basically, what you could see through the presentation, if you go a bit deeper, is that also producers of analytical equipment like Shimadzu should also, especially in pharma, work closely together, A, to understand the needs, B, to develop the support and instruments that can support the needs, and C, when and if the instruments get installed, also work along them. So when the instruments get installed, the cooperation begins. That's what I got.
Marina-san?
I totally agree with you. And in my opinion, I felt the communication is most important to make things better for everyone, attended here. And without your information, we cannot think about what we can do as a manufacturer of the analytical instrument. And today we have learned a lot of things, what our customized thinking, and this is a great input. And we would like to talk more about how we can do to improve the situation. So we would like to continue this kind of communication more with the customers. Thank you very much.
Thank you. Dr. Thomas?
Well, I think we have learned today the analytical power is here. We have to use it. What we still have to do, we have to merge that with the biological knowledge to get the added value that this analytical power is promising. And I think it's needed that we work together in different disciplines much, much closer across countries and across disciplines to really utilize that power and intelligence.
Thanks.
Thank you, Dr. I think I'm the last, so all the things I need to say is already said. But just to sum up, I think, of course, I was here at the first summit, and today is the 7th summit, and I've been into a few. But I must say that when we first started, it was more a regional summit. But today, I think it's truly a global summit. And I think the other thing I observe is that Prem has not aged a day since the first summit. Thank you.
Oh, thank you. Thank you for those comments. Do we have time for maybe one question from the floor? Well, everybody wants to leave, looks like. Well, if there are no questions, thank you very much to this very learned panel over here for sharing your insights with the audience. And to the audience, thank you very much for the patience to listen to us in this final session.
Thank you very much, panelists. Thank you very much, Dr. Satish, for excellent moderation and making this session interesting. And ladies and gentlemen, this was a final session for today. Thank you very much, all the speakers, for your time and your efforts to share all the information. Also, the panelists to share their views. And we are very sure that definitely Pharma will be growing and our companies will be growing together. And with that note, we would like to have some appreciation for our speakers and the panelists. And for that, may I request Maruyama-san again on stage to give away some small token of appreciation to all our speakers. So I will call upon the speaker, please come and stay on the stage after you receive the plaque, okay? So we will start with the first session first. Mr. Indrajit Bose from India. Dr. Jimmy Yen Chu Lin from Taiwan. Dr. Jimmy, are you here? Okay. Dr. Kavita Vengurlekar. Okay. Dr. Baoning Su from China. Dr. Baoning Su. Okay, maybe next one. Dr. K. Muralidharan from India. Dr. Christopher Welsh, next. Please stay on the stage, speakers. Speakers, can you please stay on the stage? And from the last session, Dr. Chikako Yomota from Japan. Dr. Kuihua Liu from China. Dr. Ravi Krovidi from India. Mr. Luciano Rosado from Brazil. Dr. Pakarong Kawano from Thailand. Dr. Pakarong is not here. So, Dr. Jonathan Watt from USA. And from the panelists, Ms. Aristiana Permata Aji from Indonesia. Please come on stage. Dr. Thomas Bieber. Dr. Paul Heng. Mr. Boru Che. And finally, Dr. Satish Lele. Okay, please stay on the stage, please, Dr. Satish. And all the speakers and panelists, please come on the stage with your plaques, if possible, for a group photo. And may I also invite Tanigaki-san, Ueda-san, to please also join the stage. Prem also to join the stage. compliance everywhere we have to follow. Okay, thank you very much. It's great pleasure to have such a global team here. Okay, thank you very much. And that's almost the end of this summit. And I would like Tanigaki-san to please come on the stage and share the closing address. Tanigaki-san.
Very good afternoon, ladies and gentlemen. I have been given the honor to have the final say in this mega event by delivering the closing address today. Thank you. It gives me great pleasure to represent the entire Shimadzu family to thank all you eminent pharma industry leaders for your gracious presence at this 7th Shimadzu Global Pharma Summit 2019. Thank you for your taking time from your very busy schedule to attend this event and making it a grand success with your estimate presence. Thank you. This year, this Pharma Summit was based on the theme of transforming the future of pharma. And I'm very happy to see that the diverse range of topics from AI to medical cannabis were discussed in great detail during this day. This truly highlights to us the impact that futuristic technologies such as analytical intelligence would have in the domain of pharma. We sincerely hope that the network you develop here help you to further grow and strengthen your collaboration across the world. This event is a reflection of our Shimadzu corporate philosophy of contributing to society through science and technology. We really appreciate the support you have provided us over the years, and we promise to provide you the best solutions that technology can offer for the next many decades to come. We hope that this event was very fruitful for you all, and we eagerly look forward to your presence at the next event. Thank you once again for everything. Thank you very much.
Thank you very much, Tanigaki-san. Thank you. Okay, so before we really end, it is very important to give a big round of applause for all the participants. Big round. Yeah, it was really great to have you, all of you here. And we definitely want to capture this moment to be with all of you So the next thing we will have is a group photo with everybody who is attending this Pharma Summit. And while the chairs are getting ready, I will just give you some important announcement for the evening, okay? So we have talked a lot about pharma, lot about regulation and other science, but now is the evening, more relaxing with sea aquarium, fishes around, and lot of more entertainment, okay? So after the group photo, you can go back to your room. You can change to smart casual, no need suit and tie, okay? But please remember, not too relaxing. You have to be at the lobby, Tower 3, at 5:50 p.m., 17:50 p.m., okay? 5.50, okay? And you will see the Shimadzu team there, okay? They will guide you for the coach. At the same time, they will give you one wristband. Now, this is the most important thing. If you lose this wristband, there will be no entry to sea aquarium, okay? So please remember to safeguard your own tag. Don't give it to anyone, okay? This is very strict that without the tag, no entry, okay? Please remember. And that's it. After that, we will have a very great evening at Sea Aquarium, okay? Now the stage is all ready. Let's take a group picture and then we are free to go. Thank you very much, everyone.
Okay, we have the Dr. Weda, Mr. Mariyama, and Tanigai-san come on stage to sit first. Well, then later we invite all the speakers to come out. Yeah. Mariyama-san. Yeah, Mariyama-san. Weda-san, you sit. Yeah. Okay, next is all the speaker and panelists. Please choose your seat. Yeah, I guess. Yeah, okay. Center. Okay, speakers and panelists, please come out. The chairs is for the speakers and the panelists. Yeah, please come out. Come on, all the speakers and panelists, please. Come, yeah, sir, please. Okay, the rest of the participants, please. Okay, the rest of the guests, please come on stage or in front of the stage. Okay, just squeeze a bit, okay?
please do not go to the side of the stage.
Okay, Gabriel, you come in front, Okay, some of you come in front, yeah. Yes, okay. Shimadzu staff will come in front, yeah. Okay, good. Come. Sano-san, come in front. Cannot see you. Come in front. Yeah. Yeah, please, sir. Yeah, okay, good.
Table move inside. Okay. The side is still